Hello, everyone. My name is Martin Plechner, and I'm currently based at the University of Queensland in Australia. But most of the work I'll be talking about today on super resolution microscopy has been done at Macquarie University in Sydney under the wings of the ARC Centre of Excellence for Nanoscale Biophotonics. One of the main missions of the center has been to build and develop tools that can peek into the nanoscale and potentially reveal how life works on the nanoscale. And that's a really big challenge. You have tools out there like electron microscopy that can image all the way down to less than a nanometer. But unfortunately, these tools are not exactly compatible with life itself. These imaging methods require special sample preparation procedures that essentially kill the sample. So the only thing you ever get is a snapshot of very complex interactions that constitute life. And it's very difficult to get the dynamics of interactions, the rules of the constantly changing game called life from these snapshots. On the other hand, side of the spectrum, you have these relatively gentle techniques in the light microscopy category that are gentle to life, but they suffered for a long time with the inability to break through the diffraction barrier, which is around 200 nanometers, which is where the bulk of how life works is. Fortunately, this is changing, and in the last decade or so, there has been an explosion of techniques that can pass through the diffraction barrier and image those tiny features in living cells. But are these any useful for biologists? Are there any real biological questions being answered with those techniques? So far, the discussion between physicists and biologists was mostly like this. Physicists, hey guys, we have this really amazing technique that can image this blurry triangle as a sharp triangle. And the biologist was like, well, that's great, but I already knew it's a triangle. What is the additional information we are getting out of that? Or at least it used to be like that in the early stages of the super resolution field, but this is changing lately, and there are real biological questions being studied with those super resolution techniques. Just to give you a sense of what is out there, I will give you two examples. STAT has been used to study the HIV virus maturation. When the virus is in the early stages of development, there is this GAG protein, which mostly aggregates at the outer shell of the virus. And when you image it with STAT, what you see is the outer shell with a hole in the middle. So that's the early stages, but as the virus matures, these GAG proteins change the position towards the center of the virus structure, and then the hole disappears. So you can use the STAT microscopy to clearly differentiate between the two stages, early and late. Another example is using STORM to reveal the inner structure of the axons and dendrites. It has not been known that there is this periodic structure in axons until we used the STORM imaging technique that revealed it. No one really knows why it's there, but it does exist. And if you know it exists, you can ask questions like, why is it there and what is its function? So anyway, the techniques exist and they are useful. Now, if you only had money to buy one super resolution microscope, which properties would you like your microscope to have? That's a million dollar question sometimes. And the main wishes are usually high spatial resolution. This is understandable because it's a super resolution microscope, right? But are you gonna go for something relatively moderate in terms of resolution improvements, like 100 nanometers at best, or do you want 1 nanometer or even less? What is the kind of temporal resolution that you require? Do you need to image deep, or are you fine with imaging at the surface? People also want a technique that has very low phototoxicity. They don't really want to kill their specimens. People want to image for a long time, so bleaching might be an issue because if it is present, it only allows you to take a couple of snapshots and you can't really image for hours or even days. Wide field of view is another item on the list. People would like to see how this thing over there in the corner of the field of view interacts with the thing over here at the other side, in high resolution preferably. Multiplexing is really important because there are many molecules or players, if you wish, in this game of life. And so far you can multiplex in color, but you can only fit that many colors into your dichroic mirror system. So it's about 3 or 4, and biologists would be interested to maybe watch 10 or even 20 different players at the same time. And biologists also don't like autofluorescence, which is the effect that you get when you illuminate your sample with UV or visible light. And that gives you fluorescence from endogenous fluorophores that you don't really want to see. You just want to see your targets, and these endogenous fluorophores are just adding some extra background that you want to remove, and that decreases your contrast. So optimally, you would like to have all of these things, right? But we live in a real world and you can only ever have a couple of those. So I think if you had a magic lamp and I was the genie, I can only give you 3 of these properties, and the rest, well, we will have to talk about, and somehow, yeah, you need to basically go for a trade-off with the rest of the properties. So anyway, that means you need to be very careful with 3 wishes you will go for. Otherwise, you might end up with a tool that you don't really want and with which you will suffer during your working hours, like those people on the pictures here. So which tool do you want? Well, that depends on what do you want to image. Is it some kind of structural information on a micrometer scale, like the cytoskeleton structure, or do you want to see interactions of organelles, let's say lysosomes or mitochondria, which is happening on the scale of 500 nanometers or less? Or do you want to go really small, all the way down to proteins, sugars, and lipids? It also depends for how long you need to image. Are you fine with short bursts of a couple of seconds or even a minute? Or do you want to image for hours or even days? It also depends how deep you want to image. And I would say that if you want to have 3 wishes, 1, image deep, 2, over a long period of time, and 3, track those tiny features in the cell like proteins, sugars, and lipids, you might actually struggle to find a technique among those abbreviations on the previous slides that suits you. So it looks like there is a need for another super resolution technique. But in order to add another super resolution microscope to an imaging repertoire, we first need to know what actually determines the resolution of a microscope. First thing that is a main player is how you illuminate a sample. Obviously, if you choose an objective with high NA, you get a smaller PSF and you get a better resolution. So the illumination is a big player. But you can also imagine that if you illuminate with a series of fringes, as you do in the structured illumination microscope, and then you do some post-processing, you can also potentially improve your resolution. Another thing is detector. How you detect light is another thing that changes the resolution of your system. In the case of a confocal microscope, when you close the pinhole fully, you can get an improvement of resolution of square root of 2. With a trade-off of worsened signal to noise ratio. But you also have different detection schemes that can improve resolution, like for example, the honeycomb detector in the ARRI scan, where you don't actually just get the intensity from certain point in the sample, but you get an image of the whole PSF, and then you do some post-processing on the PSF, which improves the resolution. The last thing that changes resolution of your microscope is the material that you are imaging. Let's have a look at that because it opens some interesting new avenues for improving the resolution of your image, potentially making your imaging system so sharp that you can see that I did not update the video to 2021 version. All right, so first let's have a look at what happens when you scan a linear material through a Gaussian beam. You have a Gaussian beam with a full width at half maximum of half the wavelength, and you're scanning a linear material through it. What I mean by linear material is that the emission depends in a linear way on the excitation. So as the particle is scanned through the Gaussian beam, all you expect at the detection side is basically a copy-paste of your excitation profile, because everything is linear in the system. You might lose some of the photons, but this will be the same everywhere across the profile. So you will just expect the same kind of profile with the same full width half maximum. Nothing changes in terms of resolution. That's for the case of the linear material. However, now let's have a look at what happens when you scan a nonlinear material through the same Gaussian beam. What is going to be the detection PSF in this case? Well, clearly it's not going to be the same as your excitation PSF because you have 3 different regions of emission. You have this gray region where you have excitation going up, and the emission is not changing at all. It's just zero. Then you have this purple superlinear region where the emission suddenly very quickly grows in a superlinear way. So the slope is higher than 1. And then you reach the saturation where you can't get more photons out of your emitters, and that's the yellow region over here. That means that your detection profile will likely depend on how you choose your excitation intensity of your beam. So let's have a closer look at what happens. First, for the case when you choose the peak intensity of your beam to be in the saturation, yellow regime. In that case, when the particle is very far from the Gaussian beam, it gives you zero emission. And then as it approaches the tail of the beam, the intensity there is already high enough to just quickly go through the superlinear regime that you see over here in purple. And then the emission quickly reaches saturation, even at the tail of the Gaussian beam. And then it stays in the saturation regime for the rest of the scan. What you end up with is the emission profile, the detection PSF in blue here, that is much wider than your excitation intensity profile. That means that your resolution is worse than the diffraction limit. And if you're obsessed with abbreviations like we are in the super resolution field, you can call it the crab microscopy. So anyway, that's not very interesting for us. The other case is when you actually pick your peak intensity of your Gaussian beam in this superlinear regime, the purple window. What happens then? Well, then as you reach the tail of the Gaussian beam, you are still basically sitting in this gray region. Your excitation intensity is increasing, but you can't detect any emission, and there might not be any emission even. So your blue emission profile is still zero, even though the excitation is going up. Then suddenly you reach the purple region of very steep growth, and you suddenly jump from 0 to something very high like this. And then everything is repeated with mirror symmetry as you scan through. What you end up with is the detection profile that is much narrower than the initial excitation PSF. You go beyond the diffraction limit, and I will call it from now on the C microscopy, standing for superlinear excitation emission microscopy. All right, so this works. The question now is, do we have particles that have this kind of superlinear behavior? And the answer is yes, they do exist. It's actually not just one single type of particles that gives you this kind of behavior. I will mention at the end of the presentation more examples, but in our studies, we concentrated on upconversion nanoparticles, which were convenient for us because we had some expertise in the group with them, and they have very interesting properties. So let's have a look how these work, how they give you super resolution. What you see here is a sodium yttrium fluoride matrix, and in this matrix you have some ytterbium and thulium ions. Ytterbium ions are there to capture the 980 nanometer excitation photons. Ytterbium has a very high absorption cross-section at 980, and it is there to catch a lot of these 980 photons. When ytterbium catches a 980 photon, it goes to the excited state, and then when it later drops back to the ground state, it can give some of its energy to the neighboring thulium ions that have a very interesting ladder-like energy structure. So every time the ytterbium ion throws the energy to the thulium ion, this thulium ion goes up a step, and when it climbs all the way up, it emits a blue photon. So that's why this particle is called upconversion particle, because you convert a long wavelength to a short wavelength from infrared to blue. So this is on its own. So this on its own would give you some superlinearity because you need 4 photons to give you an emission. So emission is going to depend on the 4th power of the excitation intensity, right? But there is more to it. There is another effect that actually increases the slope. So slope 4 that you would expect due to multiphoton absorption has some additional boost from the effect that I'm going to describe. And that's this, sometimes called energy looping. The rate of transition from the ground state to the first excited state, or the likelihood of getting there, is quite small compared to the likelihood of getting from the first to the second state for the thulium ion. You can see the likelihood by the thickness of the arrows here. So initially you have a thulium ion, which is quite unlikely to go to the first step, but once it gets there, it has a very high likelihood of getting to the second level. While it's sitting in the second level and there is no 980 nanometer photon pushing it higher up, it can relux down. And as it goes down back to the first level, it can give some of its energy to the neighboring thulium ions. So now we have two that are already in the first level here, and they again have a very high likelihood of going to the second level. And if there is again no 980 nanometer photon around at the time they drop down and give their energy to another two thulium ions, so altogether we have four thulium ions sitting at the first level where you have very high likelihood of going higher. And this spreads like a virus through the system. So people call it avalanche-like or energy looping effect. And then that pushes the steepness of your slope and leads to a very sharp growth of the emission as a function of excitation. Okay, cool. So it's multiple photon absorption and energy looping that contributes to high slope. Okay, so we have measured this excitation emission profile for particles that were doped with thulium ions at 8%, and we got a slope of about 6.2. If you have 4% thulium doping, this means that the average distance between the thulium ions is quite big, and then the slope drops to 4. The reason for this is kind of similar to COVID distancing rules that you see all over the world. The thulium ions are simply too far apart for energy to jump between them, similar to the virus struggling to jump between people further apart. In that case, the thulium ion is just sitting on second level with no way to share its energy with neighboring ions, and you only get the effect of multiphoton absorption. You don't get the effect of the energy looping. But because we were after as high a slope as possible, because that improves the resolution, we used 8% doping. Anyway, so how does it work for imaging? What you see here at the top right is the reference image with the gold nanoparticle imaged in our system, which is a linear emitter. You basically have a light scattered back from the gold nanoparticle, which is proportional to the excitation intensity in a linear way. And this is our base that defines the diffraction limit without fancy materials. So we compare to that. And then we pick up our excitation intensity in the saturated regime. So we expect worse resolution. And indeed, this is what we get when we image the UCNT particle at this saturation power. Then we switch the power to a high slope regime. And then in this regime, you would expect high resolution. And indeed, that is what you get. You get an improvement of resolution of roughly 2 or even a bit better. And this improvement is given by the slope of the excitation emission curve. The resolution improves roughly by the square root of the slope. So we get very nice lateral resolution improvement. But also, and for free, you get an improvement along the axial direction as well. And that's quite unique among the super resolution techniques, because you usually have to do a fair bit of engineering with your optical system to get improvement in Z as well. Here, because the improvement is material based, you get it for free. So that's nice. Okay, it works. I think at this point, it would be fair to compare how these particles with superlinearity behave in different categories compared to standard probes, because you have many requirements from biologists and resolution is only one of them. So these particles are pretty good in terms of photostability. They essentially don't bleach. We had to park a very high intensity beam on the particle for a couple of hours to bleach them to 50% of the initial emission intensity. So that's very stable. They can also be excited in the near infrared, which is unique for super resolution techniques. There are not many out there that can work in near infrared. And near infrared is very useful for biology because you have less scattering and absorption in tissues, and you also don't get this autofluorescence that I mentioned before, which appears when you excite at UV or visible. So you get a better contrast. Unfortunately, these particles are quite big. And that means that the behavior of proteins that you study might change because there is this huge particle attached to it. This is happening even with standard probes, but these particles are much larger, so it's expected that the behavior of targets will change more. Well, imagine someone is studying what you do on everyday basis, and he wants to study you by watching a bus that he attaches to you. Of course, he's not going to find out your average daily activities because you'll be struggling with the bus being attached to you. It's similar with the proteins. The particle is huge, and the reason it is huge is because the quantum yield is quite small for these particles. The quantum yield is only 2% compared to 80% for the standard probes. So in order to see the particles, we need to pack a lot of emitters in to be able to see enough light from them. The size is also a problem for the linkage error. That is the difference between where the target is and where the probe is. And because the particles are bigger, the radius of uncertainty is 1 order of magnitude higher than for standard probes. Lifetime is a bit of a tricky one. Particles have a long lifetime, which means the imaging speed is slow, but at the same time, you can tune the lifetime and you can also change the colors of these particles. I mentioned blue emission, but you can have different emission colors, and that means that you can multiplex not only in the spectral domain color, but you can also multiplex in the time domain. You can look at the decay times of these particles and differentiate between different particles based on lifetime. So you can watch more types of the targets than before with these particles. And in terms of light intensities that you need to get to super resolution, the working intensity bands are huge, but I would say we can do better than the standard probes at the moment. And because we work in the near infrared, we are less phototoxic, and also we have less scattering and absorption. So in terms of light intensity, I think we're pretty good. Anyway, so we did image in fixed neuron cells and microglia. What you see on the picture on the left here is the Alexa 647 stained cell, to give you an idea where the nucleus is and where the membrane is. And then we tune the excitation power of the laser so that we roughly get a diffraction limit of a given system for these particles. That's the top middle image. And that's what you get at the diffraction limit. And then when you turn the power to be in this very steep slope region, the resolution improves, as you can see on the top left, and quite significantly, both in the axial direction and in lateral direction. So you can see on the images, which you can see on the images at the bottom. The really cool feature of this technique is that in order to get better resolution in the subdiffraction regime, you need to decrease the excitation intensity. And this is going against all the other super resolution techniques out there. For the other techniques, you always need to increase the photon budget to get better resolution, or you need to take more pictures, like in the case of structured illumination, to later on reconstruct an image with higher resolution. More pictures means higher photon budget. In our case, all you need to do to get better resolution in subdiffraction regime is just to turn down the knob on the power of your laser and you get a better resolution. So in this case, we went from 14 to 1 milliwatt per micrometer squared. We turned down the power 1 order of magnitude and improved the resolution at the same time, which I'm really quite excited about. That's pretty unique. Apart from the super resolution linearity, you can also do other super resolution techniques with these particles. One example is the STED, stimulated emission depletion microscopy, for which you need probes that can be in an on and off state. So the on state is easy to understand for these particles. Basically, it happens when you climb the ladder all the way up and then you get a blue emission. The off state happens because you have this virus spreading in your system and there are a lot of particles on the second level, say infected. So you get a population inversion, and then when you shine 808 nanometer laser that matches this transition, you get stimulated emission, and you can basically cut the ladder in the middle so that the thulium ions can never go fully up the ladder, and you don't get a blue emission anymore. So that is your off state. The way you do STED imaging is you align two beams, one Gaussian 980 nanometer for excitation, the other one LG with singularity in the middle at 808 nanometers for depletion. You put them on top of each other, and then as you scan the particle through co-aligned beams, the particle will be in the off state on the outer edges of the beams, and only when it passes through the center, it is going to be in the on state. And that means that the resolution, the emission profile, will be much narrower than as it would be normally just with a 980B. We have done STAT imaging as well, and we reach pretty good resolution for the near-infrared super resolution technique, at least, all the way down to 70 nanometers, albeit at powers that are 10 megawatts per centimeter squared. The thing is, again, it's up to the end user in the end what are the requirements for the given experiment. So if the maximum resolution is what you need, go for STAT, but then you need to pay by relatively high excitation power, which can be phototoxic for your cells. But if you're happy with relatively modest resolution improvement, you see that you can achieve 200 nanometer resolution at powers of 2 orders of magnitude lower using C method. So you might be happy with that. So it's up to the end user, and actually these two techniques, the C and STAT, can be combined in a nice way. You can see here that whenever the C mode is used, both the axial and lateral resolution improves, even in situations when you combine C with STAT. The price to pay for this increased resolution when using C is lower signal to noise ratio, but at the same time, excitation power drops significantly. So again, it is about trade-offs, and it is up to the end user which mode suits their application the best. I promised you that I will mention other superlinear emitters that are out there, and there are a couple. There are papers reporting superlinear behavior in nanodiamond particles, gold nanoparticles, and quantum dots. In most of the cases, it is very difficult to get these particles into the superlinear regime. In the case of nanodiamonds, you need 3 different lasers to get them into that regime, whereas in the upconversion nanoparticle case, the only thing you have to do is change the power of your laser and you get them there. So it's much simpler in that case. We'll see in the future, there might be some changes in how the other materials are designed, and we might have more choice in that sense. There has also been some progress in designing upconversion particles with higher slopes by Emory Chan's group recently. They reported slopes of up to 15, giving the resolution another boost, albeit at integration times that are on the order of 500 milliseconds per pixel. Again, trade-offs that have to be paid. So if you want to know more, please go and check these papers. Anyway, I think that's enough for me. I would like to thank the whole team, which was truly multidisciplinary. We had some people working on the optics, materials, and the bio team, which is still kind of trying to figure out how to go from the situation. We have a blurry triangle, we see it better. What are some real biological applications with this? So yeah, anyway, I would like to thank all of these people and also all the institutions that were supporting us to do this research. And with this, I would like to thank you, and I hope to see you during the discussion time.